So this is one of those important macronutrients with high phosphate concentrations shown in green here. And typically where there's high phosphate concentrations, there's also high nitrate concentrations. So you can think about the regions shown in green here as those that have high concentrations of macronutrients like nitrogen and phosphorus. The dots each are showing locations where somebody's done one of these bottle incubation experiments where they've manipulated the amount of nutrients available to plankton, and then they've observed their responses. And so what you can see here are general patterns where we've got iron, experiments where iron was limiting, shown in red. And you can see those throughout the Southern Ocean around Antarctica, and then up in the North Pacific as well, as some of these areas, as well as in some of these areas where we know that there's upwelling of nutrients. And then in contrast to that, we've got these regions in the North Atlantic, for instance, where nitrogen is the primary limiting nutrient, and that's what's shown with these green dots here. But what we can also see in this figure is that we're accumulating all these data points to show us that it's not just iron and nitrogen that impact the growth of phytoplankton. We're seeing that there are other nutrients, other micronutrients, just like our graph of supply and demand suggested, that can impact phytoplankton growth. And for instance, I'll highlight some experiments with manganese and with vitamin B12 in the Southern Ocean, suggesting that these nutrients are also important for phytoplankton growth. So if I had to sum up what the work in my lab has sort of accomplished and helped scientists understand, is that we've been able to contribute important information that's leading us to this conclusion that simultaneous control of ocean biomass production by multiple micronutrients is widespread and is globally important. So this is how I start to summarize the work that we've done in the lab so far. And I'll give you one example today, and we'll talk through this example and why I think it's important. So this story starts off with phytoplankton and iron, but we also need to think about bacteria, or heterotrophic bacteria. In the Southern Ocean, iron is limiting, as I've explained. And so those phytoplankton compete with bacteria to acquire enough iron to support their growth. It gets more complicated than that because, of course, these phytoplankton are producing carbon that these heterotrophic bacteria can use as their fuel. It's more complicated than that because these bacteria are actually the only source of vitamin B12, that other critical micronutrient that supports the growth of phytoplankton. And so we've got this complex series of interactions where there's both competition and perhaps cooperation in the production and consumption of micronutrients that leads to that solidifies these interactions between bacteria and phytoplankton as important for controlling the growth of phytoplankton. So in this instance, we've seen that one micronutrient can impact the production of and demand for another. So by increasing the availability of organic carbon, phytoplankton can increase the growth of these bacteria that supports the production of vitamin B12 that can then be used to fuel more phytoplankton growth. So this is a pretty complicated set of microbial interactions. And what we really want to understand is how does this microbial drama play out in the ocean? Where is it important? And what do we need to know about it in order to predict what's going to control phytoplankton growth in the ocean now and into the future? So in order to explore this a bit more, I'm going to take you down to the Southern Ocean. And before I do that, I want us to understand a little bit more about how important the Southern Ocean is. So winds in the Southern Ocean, prevailing winds cause upwelling around the Southern Ocean. They bring deep water from deep in the ocean that has lots of nutrients and CO2 stored and built up in that deep water. They bring it to the sunlit surface ocean where it can be acted upon by phytoplankton. Those phytoplankton and bacteria, of course, grow, and the phytoplankton consume carbon dioxide that's brought up from the deep ocean. And they also consume nutrients there. So the amount of phytoplankton that grows in these environments in the Southern Ocean are really important because that shapes how much of these leftover nutrients are then available to be exported to the rest of the ocean to fuel phytoplankton growth there. And it's also vitally important for determining how much CO2 ends up in the atmosphere. As I said earlier, this amount of CO2 that phytoplankton consume is critical for determining how much CO2 is stored in the atmosphere versus moved down into the deeper ocean. Okay, so what we want to understand a bit better here is how do multiple micronutrients and microbial interactions impact Southern Ocean productivity, and how will these interactions change in the future warmer Southern Ocean? So those are our two primary questions for now. So in order to help us understand or start to get at the answers to those questions, I'll take you to the coastal Southern Ocean. And in particular, we're going to go to McMurdo Sound. So McMurdo Sound is found in the Ross Sea here, which is in the New Zealand sector of the Southern Ocean. And it's home to vast quantities of phytoplankton growth and really scenic vistas. So as part of this sampling, we start off our journey in New Zealand, then we fly down south to Antarctica, and we operate out of the US McMurdo Research Station. We can take helicopters out to the sea ice edge, and we can do some sampling at that sea ice edge where we measure the concentrations of metals, we examine phytoplankton and their growth responses, and we can measure all sorts of different kinds of biomolecules to help us understand what those phytoplankton are doing in this area. It's important that we're careful about protecting our samples from dust contamination, because these trace metals, since they're required in such low concentration and they're available in seawater in such low amounts, it's very easy to contaminate for them. So we have to be very careful with our work in order to not contaminate our samples with these metals that we're trying to study. So we can do these kinds of bottle incubation experiments that I described before. We can do this across a time series. So we went to the same location at the sea ice edge once a week for 4 weeks. And over that time, we conducted experiments and we collected lots of biomass in order to measure the response of the phytoplankton communities. And what I'm showing you here on December 28th, our first example, we did this bottle incubation study and we saw that none of the micronutrients that we added made much of a difference. None of these samples were different from our control, and the amount of chlorophyll there, so this is a proxy for how much phytoplankton there was remained about the same. But as we progressed through our season, we could see that this changed. And when we did our experiment later in the season, we could see that when we added vitamin B12, or when we added iron, or when we added the micronutrient manganese, we could see increases in the amount of phytoplankton growth with the addition of each of these micronutrients. So this was remarkable to me. I was not expecting that all 3 of these micronutrients could make a difference in the growth of these phytoplankton from the same location. But nonetheless, it was true, and we saw that multiple micronutrients simultaneously control the growth of phytoplankton in this area. And our question then was, how do phytoplankton respond to this transition in micronutrient limitation? So in order to ask that question, we could use a few different approaches. But for my group, what we really like to do is measure proteins, because proteins are how cells interact with their environment. They're the agents of chemical transformations inside cells, and they're how cells sense things, and they form a large portion of the cellular resources. So understanding protein expression patterns are really important, and they can tell us a lot about how a cell is interacting with this environment. So this is our central dogma, and of course we could be measuring DNA that could tell us what the plankton could possibly do, but in this case we spend a lot of time developing approaches to accurately measure what these cells are doing at a phytoplankton at the protein level. So in order to do that, we typically use a technique called mass spectrometry. This allows us to measure the proteins in concentration and identify those proteins so we can hypothesize about what their function is. In order to do this, we take our samples from the environment or from phytoplankton that we grow in the lab, and we extract the proteins from those cells. We chop those proteins into small pieces called peptides. And these peptides are amenable to mass spectrometry and to separation by liquid chromatography, which I'm showing you here. So this separates those peptides into less complex fractions and then puts them into this mass spectrometer, which detects their mass to charge ratio. And then it breaks those peptides down into smaller pieces. And the pieces that those peptides break down into are indicative of the sequence of that protein. So by doing a lot of informatic processing on the mass spectrometry data that we acquire from this instrument here, we can identify which proteins were present in the sample, how much was there, and then by looking at information about what we know about those protein functions, we can hypothesize about what that means for the phytoplankton cells. So I'll show you some of the data that we gather from this field sampling expedition. So I'm showing you here the change in protein concentrations from week 1 to week 3. So remember, week 1, we know we've got no micronutrient limitation. And then as time goes on, we run into micronutrient limitation. And so the proteins that are in this corner of the plot, those are the ones that are much more abundant later in the season when we know we're experiencing micronutrient limitation. So the exact identity of all these proteins doesn't matter, but I just wanted to illustrate for all of you that we can detect big changes in phytoplankton protein expression from these field samples. And we can use a lot of different approaches to try to understand what those patterns mean a little bit better. And for now, I'll show you some specific patterns that help us understand vitamin B12 and iron cycling in this system. So to start off with vitamin B12, if we want to understand what the phytoplankton in these samples are experiencing regarding vitamin nutritional status, or if they're starved for vitamin B12, we want to look for proteins that are highly expressed when these organisms are experiencing low concentrations of B12. So I'm showing you some data from some cultures that I grew way back when I was a graduate student. And we can see that the concentration of these two particular proteins, MET-E and CBA1, they increase in concentration quite a bit when these phytoplankton are starved for vitamin B12. And we know that this is because this enzyme here can help reduce the amount of B12 or cobalamin that each cell needs. And that's this one here, CBA1, it helps increase cobalamin acquisition. So it's involved in B12 uptake from the environment. So we know that when cells are B12 starved, they're working much harder to take up vitamin B12, and they're working hard to replace their requirement for that micronutrient. So what we can do now is look in our protein data for these particular enzymes. And that's what I'm showing you here. So MET-E is shown in gray, and CBA1, this B12 uptake protein, is shown in purple here. And what you can see is that both of these are increasing as we move through time. So this is evidence that our diatoms that we have in our samples here in the Southern Ocean are experiencing stress and responding to this low B12 that our bottle incubation study had suggested that they were experiencing. So we know that these phytoplankton are rearranging the proteins that they're making because they're experiencing stress due to low availability of vitamin B12. And we can see something similar for iron. So now I'm showing you in black the concentrations of a protein that is required for iron acquisition. And you can see the same pattern where we've got low expression of this protein that's required for iron acquisition at the beginning of our season. And then as we move into what we already suspected was micronutrient limitation, we see much more production of this iron stress protein, this protein that's involved in iron acquisition. So what this tells us is that these phytoplankton cells are rearranging the proteins that they're